after the talk. And if you have more questions, if we have time at the end of all the presentations, you can ask. Please keep your microphones muted, and also please sign in and use the email that you're going to vote with later at the end for the sign-in sheet, because we only count the votes from the emails that are already on our sign-in sheet. Let's start. So first in advance, I should say, I'm trying to pronounce everyone's name correctly, and if I say it wrong, I apologize. So first we have Tejasri Anuboina from George Mason University, and their volunteer mentor is Karsten Siller from University of Virginia. We can start.
Can you guys hear me?
Yes.
Okay.
And I forgot to mention, please check the chat. When your time is 1 minute remaining, we're going to give you time warnings so you can know that you should wrap up.
Okay. So hi everyone, my name is Tejasri and I'm a research assistant at George Mason University. And this is part of my ongoing research on anomaly detection. So anomaly detection is used in many fields like finance and healthcare, but my work focuses especially on human behavior. So I study on how people move across space and time and identify individuals whose movement patterns deviate from normal behavior. So identifying these anomalies make emergency response faster and evacuations more efficient. So coming for the algorithm overview, the dataset I work with contains agent ID, which acts as unique identifier for each person. We start with raw trajectories and then clean them and convert them into meaningful features. The model learns using three key features: knowledge graphs, loop metrics, and density metrics. So the knowledge graphs, before that, let me quickly explain what an ontology is. So it's a blueprint that defines how concepts like place, time, and person relate to each other. So using this ontology, we create knowledge graphs for each person. We then measure how similar one person's graph is with everyone else's graph using Jaccard index. So knowledge graphs gives us population level difference, and then the loop metrics. So this compares a person's current behavior to their own past. For example, a normal routine loop looks like home, office, and back home. If a pattern changes, skipping work or taking a new route, it signals a behavioral shift. So with loop metrics, we can understand personal level changes. And then we have density metric. So this measures how a person moves related to their surroundings. So if someone often travels through low level traffic or isolated regions, it indicates deviation from typical crowd behavior. So loop metrics, so the density metric uses spatial context difference. All these features are combined in an ensemble machine learning model and trained to detect anomalies. So this approach has improved the accuracy of anomaly detection by 10% compared to the baseline models that are already existing. And the question, why HPC? So working with data from over 100,000 agents across 75 days, we are dealing with billions of data points. So we use high performance computing to process data, compute similarities, and train models in parallel. So reducing work from days to just hours. So once the model is trained, we can apply that in the real world scenarios, such as early risk detection during disaster evacuation. So by spotting people whose movements deviate from the main evacuation flow, we can quickly identify those who may be stranded or in danger. And the next one would be evacuation path optimization. So abnormal trajectories also reveal problem areas, congested roads, or slow response zones. So helping planners design safer and more efficient evacuation strategies. Thank you.
Thank you so much. So if anyone has a quick question, please mention. Okay. So let's move on to the next speaker. Next speaker is Chandra Sarkar from Virginia Tech, and their volunteer mentor is Lishan Young from George Mason University.
Should I start? Can everybody hear me?
Yes.
Okay, thank you. Okay, good afternoon, everyone. I'm Chandra. I work as a PhD candidate in Virginia Tech, and today I'm going to talk about discovering bacterial lineages from bird lice. So before I start, I want you to think about any annoying experiences you may have had with head lice, bed bugs, or ticks on pets, right? So these parasites are not just frustrating, but they also come with a lot of medical and economical costs, especially in livestock management and in agriculture. In my talk today, I'll focus on lice, even though my research can extend to other parasitic insects. Now, have you ever wondered how lice or ticks manage to thrive on such nutritionally poor resources like hair, dry skin of scalp, fur, or feathers? Well, turns out that the key is their gut bacteria. So next slide, please. So this brings me to why I study, and the reasons are twofold. In humans, our gut bacteria helps us with digestion. In lice and in other insects, these gut bacteria supply essential nutrients like vitamins and amino acids, which help them survive and reproduce. When these bacteria are experimentally killed, the lice dies or stops reproducing or growing. The insects and the specific bacteria have evolved together for millions of years and formed an extreme form of interdependence. So a possible application of my research is selectively targeting and killing of the bacteria to develop new avenues of pest control and overcoming current pesticide resistance problems. Second, my work contributes to microbial surveillance. Over 60% of new infectious diseases, including pandemics like COVID-19, have wild animal origins. So understanding what microbe exists in the wildlife is critical for predicting and preventing disease emergence. In my work, I study over 1,000 lice samples from different wild birds across the world from various countries, one of the largest surveys of its kind. To do this, the whole lice is crushed and the DNA is extracted from it, and the chemical DNA signals are then converted into computationally readable sequences. I use various software on HPC to filter the bacterial DNA only, which is often just 1 to 5% of the total data. The parsing and filtering requires extensive parallelization and storage capacities, and currently the analysis exceeds 50 terabytes, and I have not even finished yet. This highlights the computational skill required for biological discovery. So far, I have identified over 34 bacterial species and their genes that shed light on not only the interdependency of insect gut microbes, but also on the global microbial diversity. To end, I want you to remember that it is estimated 90% of bacterial species on Earth still remain unknown. And these methods can accelerate their discovery and help understand the microscopic world that shapes ours. Thank you.
Thank you, Chandra. So let's move on to our next presenter if anyone doesn't have a question. We only have time for a quick question. If you have, please.
Could I ask a quick question?
Yeah.
Go ahead.
I was just curious, what is the pipeline? What sorts of software do you use? We're starting to do a little more on campus here, and I would just like to hear some of the things you use.
Yeah, so currently the main challenge is to filter out the bacterial DNA from the host lice. And so we have been using something called the biobakery, which is a neat data, and it filters out the host DNA using bowtie and then filters out the parasitic. And then I'm using some reference-based assemblies to already known insect bacteria to kind of get the DNA and then predict the gene with PROCA and MIGA, microbial genome atlas. Yeah.
Thank you.
Okay.
Okay, thank you. So let's move on to the next presenter, Xiaorong Shan from George Mason University, and your volunteer mentor is Hadil Al-Bahar.
Hello everyone. My name is Sherry Shan. I'm a PhD candidate at George Mason University. I'm studying environmental health engineering. So today I'm going to talk about a breast cancer detecting breast cancer through airborne biomarkers. Next slide, please. So imagine you are doing your routine physical exam and your doctor tells you that you might have breast cancer. What happens next? You schedule a mammogram, but the wait is long, the cost is high, and the procedure can be really painful. Every year, more than 2 million women worldwide are diagnosed with breast cancer. For many of them, early detection could save their lives, but traditional screening methods are expensive, uncomfortable, and not easily accessible. Now, imagine if we could detect breast cancer earlier, faster, and without pain by simply just analyzing your breast. So when cancer cells grow in your body, they release chemicals called volatile organic compounds, or VOCs, that escape through our exhale air. In my research, I analyzed thousands of breast samples collected from women across the US, some with breast cancers and others who were healthy. To handle this massive data, I used a high-performance computing cluster at George Mason University, also called ORC. With parallel computing, I can cut the processing time from months down to just days or even hours. From hundreds of chemical compounds, I identified 5 key compounds that can tell the early stage breast cancer apart about 81% accuracy. These findings are scientific foundation for a new way of screening using what we call a nose-like sensor made 6 to 8 mini sensors, each designed to respond in specific chemical patterns in breasts. But turning this idea into reality is not easy. The breath we exhale is full of changing signals, so building a sensor that can listen to the raw one is essential. That's why collaboration is important. The next step is to bring data engineers, clinicians, and data scientists to design and test a portable electronic nose that can analyze real breast samples in hospitals and community clinics. Because if a single breast can tell a story of disease, then one day it could also tell the story of life. Thank you.
If anyone has a quick question, please speak now. Okay, so let's move on to the next presenter, Adriana Valentina Fares from George Mason University, and their mentor, Valentina mentor is Xin Mei from Jefferson Lab. Please go ahead.
All right. Well, hi, everyone. I'm Valentina Farias, and I'm a fourth-year PhD candidate in transportation engineering at George Mason University. I expect to graduate in August 2026, and I'm interested in career opportunities that connect data analytics, transportation, and AI. I'm going to introduce a project focused on how we can use connected vehicle data to improve how regional travel models estimate real-world travel behavior. So to give a bit of background, transportation agencies have traditionally used household travel surveys to obtain data for planning models. A small number of households are asked to record where they went, why, and when, usually through a phone or online survey. Those responses are then scaled up to represent everyone in the region and are used in what's known as the four-step transportation planning model. This model estimates how many trips are made, where people travel, what modes they use, and which routes they take. But these surveys rely on people's memories of their past trips rather than precise data. They're also expensive and only done every few years, so data could be outdated and they don't always capture rare events like a global pandemic. So our project offers a new approach. We use 1.79 terabytes of connected vehicle data, which represents millions of trips across the Richmond metropolitan area. Each vehicle sends GPS signals every 3 seconds, and from these, we identify trip origins and destinations and link them to the traffic analysis zones used in planning models. The challenge, as shown here, is the size and the complexity of the data. Each day represents roughly 43 gigabytes of data for a period of 170 days. Once extracted, we identify origin-destination trips and analyze them in ArcGIS and Stata. Next slide, please. But before running those analyses, we had to clean the data. So we removed abnormal trips, such as those with long stops in the middle, incomplete trips, or unrealistically long travel times that likely represent regional trips passing through Richmond. Our early findings make sense. Both trip distance and travel time show two clear peaks, one in the morning and another in the afternoon, matching daily congestion periods. Trips near downtown Richmond are shorter, shown in green in the map on the right, and those that are further out in the suburbs are longer, shown in red. Our next step is that we'll compare the observed routes with those assumed in existing planning models to see where they differ and why. So looking ahead, this research can help decision makers make better understand how people actually move through their cities. And it could also lead to better forecasting, less congestion, and more efficient, safer transportation systems. Thank you.
Thank you, Valentina. If anyone has a question, please ask. Okay, move on to the next presenter, Emily Euler from James Madison University, with my volunteer mentor, Supraja Naraharisti from George Mason University.
All right, can you hear me?
Yes.
Okay. Hello, my name is Emily Euler. I am a junior chemistry major at James Madison University, concentrating in ACS materials chemistry. And today I'll be telling you about my research on modeling isomeric butanol reactivity on TiO2 gold 111 inverse model catalysts. So you may be wondering, what is an inverse model catalyst and why do we study it? Well, a promising route for fossil fuel-free plastic production involves the dehydration of biomass source small alcohols over inverse model catalyst systems to alkenes. So from here, we can see this top picture represents biomass, and that can come from agricultural areas, forests, livestock residues. That can be turned into 1-butanol, and in turn, that can be dehydrated to form 1-butene via the loss of water over Ti3O6 gold 111. So that is the inverse model catalyst system that I use in my research. And our experimental studies showed that 2-butanol on the bottom left formed both the dehydrated product butene and the dehydrogenated product 2-butanone, whereas 1-butanol selectively formed butene, the dehydrated product. So I use supercomputers at Argonne National Laboratory and a plane wave density functional theory software, JDFTX, to look at adsorption energetics and intermolecular interactions occurring on the surface to see why these reactivity and selectivity patterns are occurring. And I use 4 nodes for my calculations with 2 processes per node and 2 GPUs, and my calculations take several days to converge. Next slide, please. So essentially how I use high performance computing is I input ionic positions that I configure from molecule visualization and editing softwares like Avogadro and Vesta. And I put them into the supercomputers, and then I run minimizations on the DFT computed atomic forces until they're below a threshold of 0.005 electron volts per angstrom. And from this, I can obtain adsorption energies based on quantum mechanics. So here at the bottom, you can see some results that I've been able to gain from the calculations into the adsorption energies. So the arrows represent the adsorption energies, and a lower adsorption energy is more favorable. So we can see with the green pathway, that's 1-butanol. The 2-butanol is the purple pathway. We can see that 2-butanol has lower adsorption energies for the last 2 steps of its dehydrogenation pathway, which indicates why we may see trends in greater reactivity, forming both the dehydrogenation and dehydration product for 2-butanol, whereas we don't see that for 1-butanol. So in the future, I plan to continue using HPC resources to study the steric effects, inductive effects, and electrostatic interactions that are all impacting how these reaction mechanisms proceed on the surfaces. Thank you.
Thank you, Emily. If anyone has a question, please chime in now.
I'd like to ask a question.
Go ahead, Emily.
Looks like a pretty big system. I would just be curious, how big is the gold surface? How many atoms total, let's just say, in the calculation?
It's about 99 atoms. So it's definitely a lot. It takes several days, like I said, about 72 hours or more to completely compute the calculations. Yeah. So they're very big systems.
On the previous slide, you had showed the parameters that you do the calculation on.
Mhm.
4 nodes, 2 processes per node, and 2 GPUs. Is there any reason why you wouldn't throw more at that? I mean, these things are typically very well parallelizable. Is this just because it's easier to run a bunch more slowly?
Yeah, we're trying to balance like computational efficiency with accuracy, and we found that this was the best parallelization method for the system at this time.
All right, thank you.
Thank you.
Thank you. So next we're gonna have Ying Fei Lin from Virginia Tech. Their volunteer mentor is Kirti Patel from Virginia Tech.
Can anyone hear me?
Yes.
Okay, I'm going to start. Hi everyone, I'm Iris, a graduate student from the Biological Science Department at Virginia Tech. When I was a kid, I wanted a T-Rex as pet for my Jurassic Park dreams. However, my parents broke the news. Dinosaurs went extinct. Fast forward, I have learned we do have living dinosaurs, birds. In an evolutionary sense, a chicken is a tiny dinosaur. So the dino networks are more accurate than we thought. How do we know? We use phylogenetics trees that describe relationships between species. The key thesis is the root, which indicates the starting point of the tree and it shows the directions of evolution. Put the root in the wrong place and you will get the wrong story. For example, here is the tree with 4 tips: dinosaur, chicken, crocodile, and lizard. If we misplace the root using dinosaur, putting the starting point of the tree on the dinosaur branch, the story flips and the tree flawlessly claims dinosaurs split off first like a distant cousin, making chicken closer to crocodiles and lizard than dinosaur, which is wrong. If we correctly using the lizard branch as the starting point, then we will recover the true evolutionary story, meaning that chicken is closest to dinosaur like siblings, crocodile is next, like cousin, lizard is the distinct outgroup. That's why placing the root correctly is essential because it can tell you very different stories. Next slide, please. Root identification is a very difficult problem, although we do have several traditional ways to do so. They require assumptions or extra knowledge that we don't always have, or they don't always work well. So we ask, can a deep learning model learn to find the root directly from the molecular data? In order to do so, we perform tons of simulations with the ground truth and use to train our machine learning model. After training it, it can place the root correctly. Our pipeline needs a lot of computational resource that are provided by our high-performance cluster, ARC. How well does it work? Across all tasks, the accuracy was always above 85%. Under the same settings, a classical baseline was around 20%, more like a random guessing. This tells us that deep learning could become a practical tool that is faster and more accurate than other traditional methods. So again, why root inference important? Because the position of the root is fundamental for asking big biological questions, from the tree of life to the public health issues, such as cancer tracking, vaccine development, and infectious diseases. Our machine learning approach is a living proof that roots can be identified precisely. Thank you very much for listening, and I'll be graduating in 2029 and open to research opportunities. Happy to take any questions. Thank you.
Thank you so much. If anyone has a question, please chime in.
Yes, I have a question.
Go ahead, Grace.
Hi, thank you, Yingfei. I find it very shocking that the traditional methods led to actually worse than guessing, because guessing you would think at least 50% correct. but in this case, it's less than half of 50%. So that sounds like it's particularly with the set of species that you're applying the traditional methods to, that's particularly difficult with traditional methods. Can you say something about those species?
So like the one that we're using, we're technically using some kind of like a simulation data set and so on. So when I'm talking about like 20%, it's like under the trees that have like 4 branches. When we have like 4 branches, we will have like 5 different points for like rooting the trees. So that's why like they make it like 20% instead of like 25% or 50%. So they are like randomly guessing like each from like each one of the 5 topologies on that one. So what happened is like we use like something called like IQ tree, which is like a maximum likelihood method to like try to like root the trees, and that's showing us that like they weren't able to tell the differences between like the tree rooting positions on that one. So they are saying that like all different kind of like tree rooting positions, they have like the similar likelihood like to claim the tree is like the correct one on that. Thank you.
Thank you so much. So let's move on to the next presenter. We're having Parastoo Bybordi from Virginia Commonwealth University, and their volunteer mentor is Anurilhi Gupta from George Mason University.
Hi, can you hear me?
Yes.
Okay, thank you for introduction. Hi, everyone. I was wondering, have you ever watched any crime TV show, and have you ever wondered how detectives catch the criminals? So I'm Parastoo Bybordi. I'm a PhD student at Virginia Commonwealth University. Today, I'm going to talk about my research focusing on using machine learning models in forensic science. So let's start. Starting at crime scene, we have evidence, and these evidence have DNA samples. So the investigators extract these DNA samples and amplify them and run them through a machine called HRM machines. These instruments slowly mix the DNA and measures the changes in difference as the DNA stands separately. So the result is going to be a curve like this, as you can see at the right. So each person has a curve as a distinct genotypes. At the bottom, you can see how this is connected to the people. So we have a bunch of suspect or individual. We gather their samples and we measure the HRM curves by comparing their curves to the curves from the crime scene. We can predict if a profile matches or not matches to the crime scene, so we can make a decision if the person is guilty or not. As you can see, these curves look very similar together, and we cannot capture the difference between them by eye, and we have thousands of temperature points. So to achieve the goal of finding the differences between these curves, we need to use the machine learning methods. Next slide, please. So we have defined 2 methodologies over here. First is peak feature extraction. We extract the features like the temperature, the height, and the area under the curve for each curves, and we can see the differences between the curves. And for the second one is functional principle analysis. We summarize the curves as a smooth function and see the variation between them. And we have 2 sets of features. We run them through the machine learning models, and at the right, you can see the result. If we have 3 different machine learning models, we have the FPCA with the highest accuracy for random forest. The entire analysis involved hundreds of HRM curves, thousands of temperature points in different DNA regions. So we need HPC here to run all these samples at the same time, training all these models. And this reduces the times that we are analyzing, and it gives us a faster result and reliable result. I hope these talks helps you to know how we can capture the criminals in the crime scene. Thank you.
Thank you, Parastoo. If anyone has a question, please ask. Okay, so moving on to the next presenter, Daniel Berhane Araya from George Mason University, and their volunteer mentor is John Gillespe from Virginia Tech.
Thank you for the introduction. Can you hear me?
Yes, go ahead.
Thank you. Hello, everyone. I'm Daniel Berhane Araya, a graduate student at George Mason University. My research focuses on a collaborative approach to identifying financial misinformation using a combination of retrieval augmented generation and externally verified sources. Next slide, please. In 2013, a hacked Associated Press Twitter account falsely reported about an explosion at the White House. Within minutes, the markets fell sharply, wiping out $130 billion from the stock market. Although the markets regained quickly, it exposed how vulnerable financial systems can be to misinformation. But it's not just about money. Financial misinformation shakes market stability, undermines investor confidence, and creates blind spots for financial regulators. Next slide, please. To address this, I designed FinVet, which combines RAG and externally verified sources to provide explainable solution. RAG is a framework that combines information retrieval with LLM generation. It first searches external documents to retrieve relevant context, and then uses that context to generate accurate and evidence-backed responses. In this architecture, initially, a labeled dataset is embedded into numerical representations and stored in a vector store, which enables semantic retrieval. Subsequently, when a new claim is provided, three things happen at the same time. First, two RAG agents independently reach a verdict. A verdict in this case contains a label, evidence, source, and a confidence score. Secondly, simultaneously, the fact-checking agent queries an external API. There's a match, then it retrieves the relevant information. Given three verdicts, the voting agent uses a confidence-weighted system to reach the final verdict. These operations are computationally intensive, involving thousands of embeddings, similarity searches, and LLM inference calls. To parallelize these processes, I leveraged Hopper HPC cluster and used A100 GPUs. Experimental results show that FinVet outperforms the best of the three agents by 10.4% and has an F1 score of 0.85. F1 score is a metric that combines precision and recall and ranges from 0 to 1. Although this work is promising, there's a number of ethical considerations that needs to be taken into account when applying the framework, such as model and data bias, as well as the impact of false positives and false negatives. In conclusion, FinVet provides transparent, evidence-backed verdict that helps foster market stability, create more trust amongst various stakeholders, and most importantly, it democratizes financial misinformation detection. This work has been accepted for publication at the IEEE Big Data Workshop on Multimodal AI 2025. Thank you very much.
Thank you, Daniel. If anyone has a quick question, please chime in. Okay, so let's move on to our last presenter, Kapi Ketan Mehta from Virginia Tech. Their volunteer mentor is Eric Walter from William & Mary.
Hello, everyone. My research is based on the domain where I combine AI components with my model predictive control theory. And the context of this research is that recently in the past decade, industry has moved to using AI more and more to control their systems. Basically, if you've seen the demos of cars, humanoids, they have been using that recently. The problem with that approach is that safety is an afterthought, where the network has done all this stuff. It has given me control inputs, and I'm implementing clipping and other approaches to make it safe. Then the problem with that approach is that we don't know what the network is going to spit out in scenes which it hasn't seen in its training data. That's where my research is situated, where I use the flexibility of AI components and the safety of model-based components. Next slide, please. So my research basically has three components where I have the planner, which is based on neural networks, which is the basic unit of the AI infrastructure. Then I have the safety awareness, which is basically constraints in my environment and the safety of my own. And then I combine those on the predictive control, which optimizes both of them and spits out a model-based safe output. The image which you see above is basically the predictive control component, which is a 66 variable problem, which I'm able to solve under 500 hertz on a local system. My hope is that, or my plan ahead is that, to use the HPC infrastructure at Virginia Tech to train the planner and the predictive controller together and implement a safe controller on hardware. Thank you so much. I want to thank my mentor, Eric.
Thank you so much. If anyone has a question, please speak. Okay, thank you all the presenters. That was fantastic. We learned a lot about your research. So we're dropping the voting link in the chat now. Please cast your vote and use the same email that you used for the sign-in sheet, because we only count the votes that are with the same email. Sorry for the delay. We had a tie and we were analyzing the numbers to see. It was close to say that. Okay, so we want to thank Dell Technologies for supporting this event. They're really generous. We have one prize for the first place, and the prize would be a laptop from Dell Technologies. The winner is, let me tell, Emily Euler from James Madison University with their mentor, Supraja Narahasti.
Thank you so much. Thank you.
Please we'll be in touch with Ivy. She's gonna get your address and information so they can send you the laptop.
Thank you. I really appreciate it.
You're welcome. So congratulations to Emily. Thank you so much for attending this event, and please attend our future lightning talks and events. We're gonna have another event in December. So please keep an eye on our announcements, and thank you so much.
Thank you, Mashi. Thank you, Ivy.
Thank you, Grace.
I
Thank you,
also, sorry.
everyone.
I want to mention that Dell, DELL, they have they generously sponsor our event, but we cannot guarantee we'll have the same price every year.
Of
We
course.
don't.
That's the
We may
point.
have something.
It
We may have
depends on the
something better.
sponsor.
Yeah, we may have something better. We may have something small, but we will have another round of lightning talk in the springtime, I think.
It totally depends on the sponsor, of course. Yes. Thanks for mentioning that. Yeah.
Thank you, Lishan.